Okay, I believe we're live. This is kind of impromptu, and we don't entirely have a focused topic, so we're just going to kick around some of the stuff about the video I did last night, inexplicably at 2:30 a.m. Got up and decided to actually listen to Smokey and Maddox pick on my. Oh shit! Hang on a second here. I have to shut that off. Could you hear that?
No.
Oh, that's good. Anyway, my audio started. So basically, I finally listened to it, and I was kind of amazed that they were listening to the whole thing, and they were saying, "Well, when is he going to have a rebuttal or a rebuttal? You know, when is he going to argue against us and stuff?" And so, and then they were claiming that they knew all about biosemiotics, and I really doubt they. I think they actually learned from my video. And they agreed with everything I said because I was saying DNA is a code. So they didn't really have anything to push back, but then they were making fun of me because supposedly I didn't have a rebuttal. Well, the purpose of the video was to open the discussion about biosemiotics. And I don't know if you guys watched it or not, but it's a real simple concept. It's called a sign relationship, and it's a ternary relationship. And it's become kind of big in biology in the last 10 years. And there's kind of a war going on about it, about the people. Some biologists they want to say it's all chemicals. Let's not get caught up in codes and biosemiotics. And then you have these other people that are basically think it's a big thing. But what they've done is they've opened a door to the den of fucking beta wolves and given them, given them verbiage that they can throw back at us. And there's a part in here where they say, well, he's proving creationism, because if you say it's a code, they automatically say, oh, then it has to be written by an intelligent, a creator. For them, that is a given. What do you? How do you deal with that?
I mean, you kind of let the script write itself. I mean,
yeah.
We already know Maddox doesn't understand anything. So, I mean, why pretend like things are going to change now? I mean,
yeah. And did you hear the part where I said AUG codes for methionine, and he knows that he read it as a start codon, and it's both. And he didn't know that. And he
Wait, really?
Yeah, he was trying to say that's only true in prokaryotes or some bacteria.
Wait, can you play that clip?
Oh yeah.
Do you have that marked? Oh my god.
That's true of all organisms, and technically it's formal methionine.
Yeah, well, it's formal methionine if it's the start codon, and then any subsequent methionine would be normal methionine. But still, it's just like methionine with like 1 or 2 atoms attached.
Right.
Yeah, it's like 2 atoms attached.
Yeah, so they can get rid of them.
Yeah.
Oh, and then they went after Dr. Dan and called him a trash car salesman or something. I don't know. Let's see. I'm trying to find it here. Not good at this. Where does he? Got it. Sometimes Macs do piss me off. Okay, AUG. Okay, I got it. So I just got to share the screen. Yeah, I'll play it for you. Because in my video, I said that AUG codes for methionine.
Yeah.
And I didn't mention that it was also a start codon because it wasn't necessary to make the point I wanted to make. So,
but John Maddox has taken biochemistry at MIT.
Yeah,
I know.
he's definitely taken course level biochemistry. Definitely. Somehow he managed to get through that. That course without learning that AUG is a start codon.
Oh, and here's
it goes from methionine.
Yeah. Okay, you want to listen?
Yeah, I got it.
Sure.
Okay. Hang on a second. I queue it up again.
Well, and here's the other thing is that AUG in
that's what I'm talking about.
Well, I'm saying like AUG.
It means start, doesn't it, Maddox?
Well, that's the thing is in eukaryotes, and not all, but the majority of eukaryotes, it means start.
No, in all eukaryotes, it means start.
It means something entirely different.
It also means methionine.
No,
it means both.
in the context of the semantics, ladies and gentlemen, and the additional reading capacity that is required for the information processing of the code.
Word salad, word salad, word salad.
Yeah, okay, yeah. I thought that was precious that he thought he caught me up on something and he fucking made a fool of himself.
Can John Maddox give a rundown of the chemical processes that describe replication? I would love to see him do that because I don't think he understands it at a chemical basis. He just knows the buzzwords about it has a start and a stop and it has all these other things that a code has, but he wouldn't actually be able to describe how the tRNA recognizes the mRNA or how the protein sequences recognize AUG as the start, and then they know to remove the formal methionine because of the formal group and stuff like that. There's no way he knows that.
No, he doesn't understand. He probably doesn't understand the function of silencer promoter and enhancer elements in that are upstream of DNA as well.
Mm-hmm. Yeah,
I guarantee you that he doesn't understand that.
He knows the buzzwords, but he doesn't actually know the chemistry. And this isn't even advanced chemistry. This is intro level biochemistry.
Yeah, this is even covered in basic genetics. I mean, they don't go into as much detail about the function of enhancer and promoter elements, but you get kind of a glimpse of what they do.
Yeah.
Oh my god, he loves ribosomal pausing and Scheindel-Garno sequences, but I would love to have him explain what a Scheindel-Garno sequence even is.
Right.
Yeah.
Because that's
it was discovered in I think bacteria amoebas.
Yeah. Well, Scheindel-Garno sequences are exclusive to prokaryotes.
Yeah, 1983. And then they found some evidence of ribosomal pausing in eukaryotes. Mm-hmm. But it's weak evidence, and there's not a lot of it, so I don't know. But anyway, ask him things basically where does the ribose hook up, and where does the phosphate hook up, and what is a phosphate? But now that we're doing this live, of course he'll go look it up, but he's still not going to fucking understand what he's looking at because he's an idiot.
No, I mean,
but you'll notice in here when he was criticizing me, he couldn't think of the word methionine.
No.
So, and I mean, that's ready at hand if you've looked at start signals and, I mean, I don't know. That's probably the only code I really know well, but we know that pretty well. So.
No, I mean, the others are when it's 8 of them are for valine, and
yeah,
it's kind of a pain to have to memorize them. I know I didn't need to when I was an undergrad, but
yeah, it isn't something I'm gonna actually memorize. But anyway, so that was interesting, and there were some other parts in here that were, oh god, what? Let's see. Oh, this one, which. He has the same styles. If you listen to him,
is he going to refute it? So he was summarizing the talking point, and now he's going to refute it, right?
I don't know.
No, I'm not, because it's true. What I was saying was true. I wasn't trying to refute anything. They're the ones that thought, "Oh no, this is a big, rebuttal." I don't know.
Apparently, everything to Smokey is a conspiracy.
Right. Yeah.
Well. Yeah, everything has to be an argument that can be won or lost. There's no such thing as just explaining something.
Yeah.
Yeah, there's no such point as people making valid points to Smokey, because that would have to mean that Smokey doesn't understand something and is willing to accept new information.
Is it your claim that Immutable has failed? Is that your claim?
Yeah.
Well, he's failed as a human being, or as
okay, Michael.
What?
Appreciate that.
Oh, yeah. You showed up, so you showed up.
Hey, that's what counts, right?
Yeah, but the
defining moment of my life.
Yeah. Well, let's see. It's my John Maddox impression. Oh. Oh, okay. That was his John Maddox impression.
Now you got to read it like John Maddox.
Yeah, I can't do that. I don't do it. I'm trying to do the the premise, literally.
You can tell when John is making shit up, when his speech just slows dramatically.
Yeah. He's still He turns up the condescension and turns down like how much sense it makes.
Right.
Yeah, exactly. It just, and he doesn't seem to notice when he's doing it either, despite how many times people have pointed it out.
Yeah, he came up with this. I was talking about biosemiotics in relation to amoebas. And what happened to StreamYard? Hang on a second, guys. I think I lost you guys. I don't know where I lost you. Hang on a second. I'm working on this. Okay, I'm back. Okay, so he came back and he said, okay, so the problem with this is just indicative of the standard atheists, like just blinders. They're completely refusing to comprehend the premise that the gradient and the different sensory mechanisms in the amoeba enable it to react, require information processing capacity that is formulated via prescriptive information requirement for those mechanisms, which enables information processing. This is the entire point.
Okay, could you make that more of a word salad if you fucking tried?
Okay, so let's
I can do better than that, like in like
For
2
someone who talks about
sentences.
semantics a lot, he sure doesn't seem to understand it.
I can summarize that as like there's prescriptive information for amoeba to have certain biological functions.
right. Wait, where the hell is the amoeba? Okay, so now that I've read what he said, I'll play it.
Was there a point? I don't know if I heard one. Was there a point?
I think this is
Well,
the
okay,
amoeba.
so the problem with, and this is indicative of the standard atheists, like just blinders. They're completely refusing to comprehend the premise that the gradient and the different sensory mechanisms in the amoeba that enables it to react require information processing capacity that is formulated via prescriptive information requirement for those mechanisms which enable the information processing. This is the entire point.
Well, that's what I was saying. Like,
you
is he
don't
trying
think-
to prove creationism here right now?
So you somehow don't think that the information like necessary is pertaining to the structure of the molecule itself.
I think he's almost saying that there has to be this prescriptive information and forethought in order for the fucking amoeba to run away from a pH gradient, right?
I don't
That's
know.
the argument, and
I
that's
mean,
stupid.
If that's the argument, that's stupid. But I mean, if he's saying that like proteins have prescriptive information from their amino acid sequences, sure.
Yeah.
That's chemistry, though. Like there's nothing overly surprising about that.
Anyway, the thing I wanted to drill home here is that they listened to that entire video and they couldn't find any way to really disagree with what I was saying because it's kind of their talking points. But from that, they're like, okay, if he agrees with us, then he must believe in God, because how could he believe in biosemiotics if he doesn't believe in God? I mean, God obviously did it, right? How else could it have happened? And it's just, and I look and I think about that. They make this, I explained it in the video. If A, then B. They don't do the then part. They never. I am not.
It's AB.
I
Yeah,
I am not compelled.
That's actually, yeah, that makes a lot of sense because they see like you're agreeing with biosymbiotics, so you think, or so they don't understand what you don't understand.
Right.
And this is what we mean whenever we say, Maddox, your arguments aren't compelling for your case. Like,
Exactly.
even if we grant that DNA is a code or whatever, who cares? That doesn't disprove anything. And this is like a textbook example of that, I guess.
Yeah, I mean, the other thing, I mean, my biggest issue with them is like you have to show that your model, if you don't even have one, has any predictive power whatsoever. And
I'm not even clear what his model is. It's almost like he's saying that there's some, like when he was talking to Top Dog Shattuck in 2 different videos, and I went over some of it this morning, and he was just like being just an abusive jerk, and he was trying to get out of him. He was talking about tRNA, and he was talking about the code, and how there's no chemical connection between the codon and the amino acid, so there's no chemistry at all there.
Oh my god!
So he's doubling down on the opposite side of that, saying that it's a code, but there's no chemistry, rather than saying it's both. And I'm pretty sure that he has something in his brain where he thinks, I don't know, spooky stuff is happening.
God has tweezers, and he sees the DNA code, and he starts doing like the hydrolysis reactions what is it, peptidate? What is the specific reaction type? I don't remember. Oh well.
Do aminoacyl tRNAs like have a binding domain, like to recognize codons? I mean,
amino acetase doesn't, but the tRNAs do. They have the anticodons.
Yeah, I mean, it's just, I'm trying to think like how the protein like recognizes its anticodon.
Oh, okay.
Yeah,
I have no idea. I'm sure Demeter could talk about that because he brought that up whenever he was talking about abiogenesis stuff.
I wasn't paying attention. What'd you guys ask?
We were talking about amino acetase. God, I'm fucking tired. I just woke up.
Yeah,
amino acetase.
Yeah.
Binding domains.
Yeah, I'm asking how like aminoacyl transferases like not understand, like how they recognize like the specific anticodon that corresponds to them.
Yeah.
Oh, you mean on the anticodon side, or are you talking about on the amino?
Well, not like how the tRNA recognizes the anticodon, or not how the tRNA recognizes the codon, but how the amino acid knows which codon or which tRNA to put.
Oh, right. Okay,
Yeah.
I think that is specific to the structure of that of the some of the loops up on top. What was it? I'd have to look at the diagrams, but I mean, they're all different, basically different adapters, and they pick a different amino acid. And there are some that aren't so good. I mean, there's some like fuzziness going on, and occasionally they can screw up or something. But we'd have to look into that in some detail. But anyway.
Yeah, I'd imagine it'd have to be like some recognition between like some part of
Yeah, it depends on those.
Between some part of the protein and some part of the codon loop.
No, I think it's the amino acid, and I think it's the nucleotides in that tRNA at the top of that loop, or not at the top of the loop, but on the
on the stem.
One of the ends. Yeah, I think. Anyway, it's just something that something I'm still studying. So anyway,
it's a minor point.
Well, would molecular alchemy know?
Is he here?
He might have.
He's actually here
at work right now. Well, he's at work right now, so he said he'd probably have to be a little bit in and out.
Yeah, but he's unmuted. Okay. He's unmuted. Okay. Oh, he is muted. So he's muted. So anyway, yeah, we could pull up some stuff on that. I've got.
It's good though. But either way, it's not magic. Like it's not.
No.
God's not sitting there with his tweezers and like putting everything together.
It's not like God is a protein whisperer.
Yeah. Protein whisperer.
Okay, brain bug is. I don't know how to do the tweetering very well yet. I'm still learning to tweeter. So I'm just getting used to Twitter. I don't know. I think it kind of sucks.
Twitter. Yeah, it's all right.
I couldn't tweetering the entire weather report. That would take me like 5 hours to figure.
No, he's saying like tweet out like when the times are.
Oh yes, okay. I'm sorry. I can do
like that. That's pretty much all I use my video or my Twitter for. Like whenever I post a new video or something.
I thought you guys were doing that for me.
Am I supposed to put whenever you live stream? At like 5 a.m. when I'm asleep.
2:30, man. You should have been watching.
If I'm your social media attendant, I expect wages, Michael.
Wages. Well, Yeah. We're making a ton of money on this platform. I'm telling you, it's just rolling in.
It's your public relations manager.
Make a Patreon.
Yeah. That would be a good idea. I just feel like it's self-promoting, and I guess that's what Twitter is for, right?
Yeah, I mean, self-promoting on Twitter is different than like going into people's comment sections and being like, "Please like my video." You know what I mean?
Yeah. Okay. I feel kind of bad about self-promoting myself.
I do too.
Prefer to self-promote other people, or promote other people rather than self-promote.
No, I get that.
I don't feel as bad like when I see how like bad like some people try to whore themselves out on Twitter.
Oh my God, fucking Maddox on Modern Day Debate. He buys like $30 worth of super chats, plus he puts it in every 20, that he's having an after show.
Yeah, like every 10 minutes, it's just spams up the chat.
Yeah, and I think he said he was buying ads or something like that too on Google to get people on his site. And it's
well, I mean, buying ads is not as bad as like going to other people's videos and being like, "Hey, please like my video," because that's, I don't know. I just disagree with that.
How the hell do you buy ads? Like, if we were to buy ads for this site, how in the hell? What would be the market channel for this kind of crap? I don't know.
I don't know.
I don't know.
Just put it in front of one of like every of John Maddox's videos and like Smokey's videos.
By the way, if people have not checked out like John Maddox's Twitter, bring some booze with you. Yeah, you'll have fun. It's kind of what you'd expect given like what he's told us so far about himself. Yeah,
I agree. Subscribe to Brainbug. But
you can see kind of like the decline in the apparent decline, let's say, in Maddox's mental facilities.
Search.
Yeah. Subscribe to.
Wait. So does this video have anything else about it?
Oh, there's a ton, but I kind of went through it and covered it all last night. But I think there's some stuff at the end. Well, there's other good stuff here when they talk to Manny Karen and go through the atrocity that conversation was. So hang on a second.
Molecular alchemy is back.
Oh, molecular alchemy. We had a question about amino acid transferase and like how it recognizes which tRNA to put the amino acid on. Do you know?
He's muted again.
Ah, damn it.
No, he's back.
That's a good question. It's been a while since I covered that.
Yeah, fair enough.
For a second.
Yeah, I was kind of wondering about that since Maddox raised it up. I was like, that's actually a fairly good question. I'm not.
Yeah, but I'm assuming it's not like an unknown pathway. You know what I mean? And it's definitely not like a supernatural pathway.
I don't think there's ghosts apparating like in and out of cells, like directing proteins where to go.
the Holy Ghost is a biochemist. When they say the Holy Ghost is inside of you, he's in each individual cell.
Well, that rhetoric that indicates that John thinks there's something kind of spooky going on has only been popping up in the last 2 or 3 weeks. And
Otangellum sounds like that too, actually.
Does he? Wow.
Yeah. On a lot of his like developmental stuff, it sounds like he thinks that God actually just directs like the chemical gradients and stuff. I don't think he understands how like development works, but
somebody should tweet this video out, and then Otangello will be here in a minute.
Otangello.
he's always there. He finds videos so fast.
Yeah, he hasn't found it today, so he hasn't come across that.
He's probably asleep. What time is it in Brazil?
That's a good question.
I think he's 2 hours ahead of us. So
it's like noon.
Yeah, it is 11:30.
Why does he go on the live streams at like midnight? That's like 2 a.m. for my guy.
It's about 12:20. Let's see, it's 12:26 in like normal Brazil, Federal District, and in Ilha de Fernando de Noronha, it's 1:26.
Okay.
I probably butchered the fuck out of that name.
Oh, the post office.
So, when it comes to specific tRNA molecules binding to specific amino acids, there's there's specificity in so they're not forming. I don't think it forms an ion. I think it forms a single covalent bond that's actually involved in like a substitution reaction to actually add the amino acid to the growing polypeptide chain. But the tRNA has specificity kind of like we talk about with enzymes with lock and key.
Mhm.
But these are flexible, so it's important to remember all this is happening in solution, in water.
Yeah.
And all of these structures, macromolecules, whether it's tRNA or polypeptide chain or a full-blown protein, they have flexibility. So there may be a low rate of tRNA molecules binding to the wrong amino acid. It's probably like so much in biology where you have kind of a statistical distribution.
I'm not sure about other chemists, but I would say the breaking or forming of a covalent bond is definitely chemical interaction.
I think there's a different amino acid, amino acyl transferase. Isn't there a different?
I was trying to remember if the tRNA actually covalently bonds to the amino acid, or if it's just electrostatic interactions or hydrogen bonding.
No, I think it's I think it might be covalent, but I think there's an amino acyl transferase for every tRNA. I think, right?
That makes sense.
Yeah, there is, I believe.
Yeah, no, reading, God, somewhere in this biosemiotic stuff, they were talking about that the tRNAs were central to the evolution of the protein. Okay, we have the codons and we have the proteins, right? And in between we have the tRNA. They were saying in biosemiotics, I think it's Barbieri, he was making the case that was central to the evolution of that codon, of that code, that it started with the tRNA. And he did a metaphor. He said that if you saw plans all over for building certain objects in a city, for building the buildings, and then you saw the buildings, would you concentrate on the buildings and the plans, or would you concentrate on the thing that built them? In other words, the tRNA is in the middle, and it's what built, what read the plans and built the buildings.
Yeah.
So anyway, he was pushing back on. It was called the. It was sort of the. What do they call it? Paradigm. They have a name for it. Anyway, in biology, kind of the central dogma, I think they called it, that it was codons and proteins on the other side. And he was pushing back on that and saying that you have to look at that ternary relationship in abiogenesis in order to really understand evolution. And that's Barbier's thing. Yeah.
It's also interesting because people who aren't chemists don't know this, won't understand this reaction, but there's a reaction which is called the Diels-Alder reaction. It essentially causes two reactive species to bridge together and form a more stable structure. But the point of bringing that up is there's some chemical reactions, like a Diels-Alder reaction, that are hard to achieve in normal chemistry. You have to use very high heats. But it turns out, if you synthetically build ribozymes just based on random sequencing, ribozymes have universal catalytic activity, and some ribozymes can facilitate that type of reaction to occur. Actually, called Diels-Alderase ribozymes. But it's interesting. The notion of ribozymes, prebiotically, is thought to be a very important aspect of how cells might have manipulated that as a type of proto-metabolism, to achieve certain high energy chemical reactions that may not have been accessible due to protein synthesis still being in its infancy.
Well, and it's surprising how few nucleotides you actually need to have ribozyme activity. I've got some papers I could share with you guys. I think it's like the minimum. If you have just 5 to 7 RNA nucleotides in a row, it's sufficient for it to fold into a ribozyme structure and actually catalyze reactions. So we're not talking about hundreds. We're not talking so many times when our opponents talk about abiogenesis, they compare it to modern life, modern molecular biology. And for example, like the ribosome, where you have, all these proteins, this huge RNA structure that doesn't seem likely to spontaneously arise. But if you only need a dozen or so nucleotides, and it's capable of folding into a ribozyme, then that can provide those functionality.
Anyway, I got a book recommendation from Manny Karen, who is in our chat right now.
Okay.
And it's biosemiotics, Essential Readings in Biosemiotics by Donald Favreau. And that's where I was reading stuff with Barbieri at the, and I think I'm on, I don't know what page I'm on. It's pretty long.
Did you already have the book, or?
No, this one I didn't. I had to get this book.
Okay.
But I have another one on biosemiotics by, and why can I never remember his name? And why don't I know where it is? Where is it? Oh, it's right here. It's open. Biosemiotics by Jesper Hoffmeyer, and he goes through a bunch of stuff. And he was picking on physicalism, which really pisses me off because they seem to equate physicalism with strong reductionism. Like reducing everything to just chemicals. And for me, physicalism is more expansive than that. That reductive approach I call materialism. And but these guys, he kind of picks on. But anyway, in this book and in other places, they very. I can read a quote here I found. Let's see. The crucial point: rejection of the semiotic nature of life. Hang on a second. There was a quote here that kind of talks about the maddoxes and the Smokey ickies. Meanings do not exist. We discover semiosis in every cell. Okay, hang on. Go ahead and talk, guys. I'll find it.
Yeah, it's also curious. A pretty interesting field I'd want to kind of read on is abiogenesis researchers trying to discover more what are called autocatalytic networks for chemical systems, which are actually quite interesting. So autocatalysis, for those that aren't aware, it's a molecule that helps promote its own synthesis. And it's actually quite fascinating. Like, there's a few videos that go over it really well. And the foremost reaction is thought to be a pretty good example of a system that can potentially be autocatalytic.
For some reason, the word autocatalytic now, I just have mentally in that time when Dragnaut went over to one of their after shows or something.
Yeah.
Do you remember that? And somebody asked him, is like, well, how do you get an autocatalytic reaction to start or something like that? And he's like, by the fact that you just asked that means that you have no idea what you're talking about.
Yeah.
Such a good moment.
Yeah. Reactions, like at least biological reactions, like in enzymes that are autocatalytic, don't really need energy to have their reactions facilitated. Whereas certain reactions, like various places in like the Krebs cycle or very early stages of glycolysis, need like cofactors like NAD, like NADH or NADPH, in order to kind of donate a proton or transfer electrons to help the reaction get going. And in the most extreme cases, I mean, you have some proteins that require, or some reactions actually that require the hydrolysis of ATP into ADP and the phosphoryl group.
Okay.
Okay, so Barbieri, which is one of the guys that wrote papers, 1 or 2 papers in that Royal Society thing that Jacques seems to think he discovered from 2016. He says clearly it was the genotype and the phenotype which had to adapt to the ribotype and not the other way around. Well, that's kind of interesting. He's
Yeah.
and then he says he compares the chicken or the egg debate about the origins of cellular organization to the problem of trying to explain the presence of a city full of objects that are assembled according to the instructions as an instruction first versus an object first dilemma, while regulating as mere instruction object intermediaries the city's inhabitants who are actually the entities that build the objects. So he's talking about tRNA as being more central and that the adapters came first and are more central. And then it goes on to say Barbieri has no intention of assigning to the ribotype any kind of supernatural or anthropomorphic agency for its role in bringing about the maintenance of this state of affairs. He concludes that the laws of chemistry are at work here, of course, as well as the laws of physics and natural selection. So nobody in that entire Royal Society group would agree with John Maddox on his if-then part, where he says, if it's biosemiotics, then God did it. So
yeah, of course not. I mean, the only thing that's kind of interesting, and it's a nuance point, is that even though like the tRNA is more essential, you do need to have like stable replication, like of whatever, like a stable and accurate replication of whatever molecule is being used to store information. Like otherwise, the genetic code like wouldn't be fully. Doesn't seem like the genetic code would be fully stable necessarily. Like if it was too prone to mutation, because otherwise you wouldn't be able to assign like a tRNA to a codon very easily if the DNA in that cell was just prone to mutation or oxidative damage or other things like that.
Mm-hmm.
You could potentially still transcribe and translate it. You would just end up with a mess of non-functional protein,
Yeah.
in all kinds of weird. The chaperones wouldn't know what to do with it. Doing a callback to the question about tRNA. How does a particular tRNA molecule bind to a specific amino acid? Like you guys were saying, like this is intro biochemistry. I hadn't looked at this stuff in like 11 years, but pulling up some literature about it. Basically, each tRNA molecule is made of a different sequence. Because of that sequence variability, you have different 3-dimensional structures. That is what allows specificity. So only certain structures can bind to certain amino acids, but it's not a direct interaction. And this is what I had forgotten. But there's a third player involved known as tRNA activating enzymes. So the tRNA binds. So the specificity actually isn't in isn't direct in the structure of the tRNA. It has to do with these tRNA activating enzymes. They have to have the tRNA has to fit in the correct pocket. That also the amino acid fits into a second pocket on this enzyme.
If I'm not mistaken,
an ATP dependent reaction to activate the tRNA.
Right. The acyl group has to be activated for it to be transferred properly,
So,
if I'm not mistaken.
But thinking about, I mean, this is a complex reaction and it's interdependent, and I can understand at first glance how people might imagine, this couldn't arise naturally on its own. But what we're looking at are modern molecular, our pathways in modern molecular biology. We actually, if you actually look at the extant organisms, what we're describing is the conventional paradigm, but it's not even actually true for all of life. There's variation in that in life that we can see today, and making the assumption that the very first protocells required tRNA activating enzymes in order for life to arise, I think it's an incorrect assumption.
Yeah, I mean, I could easily imagine that like there might be like some archaic, like some archaea or archae that way down deep in the ocean might be able to achieve like protein translation without needing to activate the acyl group. They might use, instead of ATP, they might use some type of inorganic catalyst to help transfer that reaction. And that inorganic catalyst might be one that's bioavailable within the environment around it.
Or it may not even follow the same steps. It could achieve the same outcome in a much more messy fashion.
Yeah, it could.
A lot of errors, a lot of misstart, false start.
Right.
So much of biology is about a statistical distribution, and steady state conditions, meaning, that you may have a whole bunch of different products, but as long as some of those products work or provide a function that promotes cell division, promotes homeostasis, you can end up proliferating.
Right.
Yep. Hey guys, I have Bible study in 15 minutes.
All right.
All right. Well, looks like this has been a fun conversation, and looks
It
like
has been.
wrap it up.
Yeah. So we covered what I think I wanted to cover. I just wanted to kind of double down, but I really would like it if some of my other teammates here would actually watch my goddamn videos.
Okay. In
What are
my
you talking about,
defense,
Michael?
you asked to start this call literally 15 minutes after I woke up.
I know.
If you're going to put out like a 20 or 30-minute video, you can't expect me to watch that in the 15 minutes I'm awake.
At 2:30 a.m.
Yeah.
Yeah. Anyway.
Come on, Michael. Let's be reasonable here.
Yeah. Okay.
I watched the first 5 minutes.
Okay. Point taken. I'm being unfair. But did you watch my biosemiotics video, which was crap?
I did.
I did watch that video.
Did you learn anything?
I might have.
Good. You're not going to admit it to me, though, are you?
Not now.
Nope. Not live.
Not live.
Okay. Good enough. Let's end this. So thanks for showing up, guys. Thank you. All three.
All right.
Yep.
It's good to show up, That's the least we can do.
That's all we can do.
Okay. Goodbye.
Hey, Speed.
Yeah.
Let me say,
Go
let me
ahead.
say one more thing. We were talking about abiogenesis, the stepwise origin of life, and the three of us were kind of speculating. But I just want to make the point that there is a wealth of experimental data that actually teases out what's plausible and what's highly unlikely. That any viewers, if they want to get into this stuff, while we're speculating here, there may actually be some phenomenal models for these exact steps or these exact aspects of life out there in the literature. Thousands of papers written on this matter.
Yeah. In fact, how many have we listed so far? We must
Quite
have
a few, like 10,000 at this point.
Yeah. Yep. We just got to go through them and figure out what's important. But
Yeah.
yeah, and I might help you with that a little bit. It's just I've been a little busy and I've been a little tired. This
I
going
know,
through
Michael.
and-
You've been a busy bee. You just gotta get you stabilized here.
I need to stabilize. I'm going to need to take like complete mental health days off at some point here, like 1 day a week, because getting wrapped up in this shit, you can I don't know.
You can do things.
May not be healthy.
Might not be.
Too much maddox would, it's not healthy for anybody.
Just remember, you've had dreams about them, so that's already a bad sign.
Yeah,
9 out of 10 doctors would recommend against Maddox.
I think so. Yeah, and Smokey, and and I'm not going to bring up the thing about Smokey and the toads and the rumors I've heard. But
Oh no.
are we still live? Shit.
Uh-oh.
Okay. I'm going to end the broadcast and we'll talk about it.
All right.
Okay.
All right.
See everybody in chat. Thank you for showing up.